Hello, my name is Mark Levitt, and I will be presenting some of the data behind the use of Ceplene as a therapeutic agent for the maintenance of complete remission in acute myelogenous leukemia. On the next slide, just to give a brief overview, Ceplene, which is histamine dihydrochloride, is an analog of histamine, a naturally occurring immunomodulator. It is administered together with low-dose IL-2 as a treatment in acute myelogenous leukemia. The dosage and administration for the proposed indication are as listed, with both drugs being given subcutaneously BID for the first 3 cycles, 3 weeks of treatment, followed by 3 weeks of rest. And during the next 7 cycles, 3 weeks of treatment, followed by 6 weeks of rest. It's already been approved in 30 European countries and in Israel, and the orphan status was granted in the U.S. in December of 1999 and by European Commission in April of 2005 for remission maintenance in AML. The target population, the estimated new cases of AML in the U.S. were approximately 20,830 in 2015, and in the EU Big Five, 15,352. In Europe, the therapeutic indication is Ceplene maintenance therapy is indicated for adult patients with acute myeloid leukemia in first remission, concomitantly treated with interleukin-2. We have learned a great deal more about AML biology, Ceplene mechanism of action, and beneficial effect in patients with AML since the EMA approval in 2008, and I will be reviewing much of this material. First of all, one has to understand that AML continues to have a very poor prognosis, and treatment really has not progressed much in the last 20 years. It's an orphan indication with a high unmet need, with relatively low 5-year survivals, particularly in elderly patients who comprise the majority of AML. Other than Ceplene in Europe, there are still no approved treatments for AML to maintain complete remission following the first CR. There's very strong pharmacological rationale for the combination. IL-2 is capable of activating and expanding T and NK cells, while Ceplene protects T and NK cells against inactivation and apoptosis, and we'll discuss this a little more in the coming slides. So the treatment is highly appealing to both patients and physicians. It can certainly maintain the duration of leukemia-free survival significantly, saving the patient further highly toxic therapy with cytotoxic agents for a prolonged period of time. It's safe with no treatment-related mortality and easily administered at home, and well tolerated with no major impact on quality of life. So it occupies a unique niche in the treatment algorithm. Just to briefly run through more of the pharmacological rationale for the combination, T and NK cell recognition of the leukemic cells and subsequent elimination of these cells helps determine the likelihood of maintaining CR in AML. T and NK cells are frequently dysfunctional in AML due to inhibition by myeloid cell-derived reactive oxygen species. And this occurs particularly in M4 and M5 AML by the FAB classification, and I will discuss this a little greater depth subsequently. IL-2 activates multiple functions of the anti-leukemic immune response, but the inhibition of T and NK cells by reactive oxygen species compromises the immunostimulatory effect of IL-2. Ceplene inhibits the enzyme NOX2 and consequently reactive oxygen species generation and its induction of dysfunction apoptosis in T and NK cells. In vitro and in vivo results have been reported in approximately 30 peer-reviewed publications. Now, this next slide is a little schematic of NOx2 complex that results in ROS production. And it's expressed by normal myeloid cells, particularly monocytes, macrophages, granulocytes. And it's expressed highly by subsets of leukemic cells in AML, the FAB M4M5 group, and also in CML and CMML. The NOx2 ROS axis mediates immunosuppression exerted by myeloid-derived suppressor cells, and NOx2 positive myeloid cells infiltrate many primary and metastatic tumors, meaning that it's not unique only to AML. A phase 3 trial was performed comparing Ceplene and IL-2 treatment for maintenance of remission in AML to no further therapy, as there was no and is no approved maintenance therapy other than today Ceplene and IL-2. So 320 patients were randomized to receive either Ceplene IL-2 or standard of care. Again, no further treatment. Were treated as I described earlier, observed for 18 months, and analyzed 3 years after the last patient. Of the 320 AML patients in complete remission, 261 were in the first complete remission. It did include all FAB subtypes, as less was known about the biology of the drug at that time. The primary endpoint was improvement in leukemia-free survival. It was a multinational trial, evenly randomized between either the drug treatment or no further therapy, and was stratified by CR status, mainly first versus later CRs, and by country. The treatment I've reviewed previously, I will not go over that again, and essentially 10 cycles were given over a total of 18 months. On this next slide, we see the data for the primary endpoint from the phase 3 trial. As we can see, there was a statistically significant improvement in leukemia-free survival in all patients. And while the numbers were smaller for the CR1 population, the gap between the two curves is even larger. And particularly in this trial, if you looked at patients less than 60, and we'll talk about this a little later, if you look at patients less than 60, the delta between the treatment and no treatment arms was quite large for leukemia-free survival. So the study met its primary endpoint of statistically significantly improved LFS at 3 years. And it was significant both for the overall population and for the CR1 population. And the Cox-adjusted models also backed up this finding. And at 3 years, 40% of the Ceplene IL-2 treated patients were leukemia-free versus just 26% of controls. As time went on, we began to understand the mechanism of activation of the histamine 2 receptor on leukemic cells and its implications. As you can see from this reanalysis that was done a few years later after the trial was finished, the FAB M4M5 group had a highly statistically significant difference in survival between treatment and no treatment, while the and so did the non-FABM2 group, although again this includes the M4M5 in that group, while the FABM2 group had completely overlapping curves. Now, what's important to understand is that the FABM2 group expressed little to no H2 receptor and thus had little to no NOx2 production. So a mature AML cell, produces reactive oxygen species, which can cause apoptosis of NK cells. Also, it can that can end up, downregulating IL-2 production. So the role of Ceplene in terms of activating the H2 receptor and downregulating NOx production, particularly in the case of AML, in the M4 and M5 subgroups that highly express this receptor, explains why we can reverse the immunosuppressive effect of the M4, M5 leukemic cells. Now, when overall survival analysis was performed initially, prior to this biological knowledge, the overall population had a trend, but not significant overall survival advantage to treatment. And the trend was a little stronger in the CR1 population, but again, this trial was not powered for overall survival and thus did not hit statistical significance. So subsequent reanalysis demonstrated, as published by Aurelius in 2012, analyses of overall survival showed trends similar to those observed for LFS, and for both the M4, M5 population. And as you can see, that neared statistical significance, as well as for the total non-M2 population. This OS trend was particularly strong in this trial in the group under 60 years old. Results in the over 60 group was not as good when stratified by FAB classification. So we have reanalyzed the data and found that the FAB M4, M5 population under 70 years old, not 60, 70 years old, in CR1 had a hazard ratio of 0.72 for OS, which is a fairly nice hazard ratio. This suggests the selected population has a good chance for success in phase 3 with OS as the primary endpoint with a limited number of patients in each cohort. I would also like to point out that this data was, we found the overall population was highly influenced by a handful of very elderly patients who did poorly. And therefore, even if we looked at the population up to 70 years old, we still had a nice separation between the curves. So to summarize, a positive trend in overall survival was observed. The median prolongation overall survival, in fact, was 64 weeks in the CR1 population, and that's without separating out the M4, M5 group. As mentioned, the phase 3 pivotal trial was not powered to detect a statistically significant OS benefit. Given the strong correlation between LFS and OS observed in the analysis of the Ceplene IL-2 data, LFS can be considered an appropriate efficacy endpoint and was accepted by the EMA. In mechanism-based biomarker-selected cohorts, the OS reached near statistical significance despite small numbers in these cohorts. So new statistical information that we've generated also suggests that a modestly sized study could succeed in phase 3 with OS as an endpoint, which the FDA has pointed out is in fact a requirement for their approval. Now I would like to take a quick look at the safety data. Overall, this treatment is very safe. it should also be noted that this is a very low-dose IL-2 administration as part of the regimen, which probably helps account for the safety. There was no treatment-related mortality. The expected symptoms of flushing, injection site reactions, headache, fatigue, pyrexia, nausea were all transient and easily managed at home. Overall, the full toxicity listing was very consistent with activation of both H1R and H2R, as well as those expected from IL-2. There was a low frequency of grade 3 or 4 and serious adverse events in treated and uncontrolled patients, and there was no major negative impact on quality of life. So, as you can see from the integrated database, even through a few years ago, we had already treated almost 1,200 patients altogether, and not just acute leukemia, with the Ceplene IL-2 combination, as we've described it. And as you can see, the non-Ceplene IL-2 treated patients in these clinical trials had, in many cases, a comparable toxicity profile with only certain ones, as I've pointed out, being significantly elevated with treatment versus no treatment. And there's also some new emerging data as we've come to understand that we can take a precision approach based on our knowledge of the H2 receptor biology as well as its distribution with AML. In a phase 4 trial requested by the EMEA after approval in Europe, more data was obtained. We saw that H2R expression increases during the course of histamine IL-2 therapy. And the higher H2R value when we see it increase, in other words, we see a nice increase as opposed to the patients where we didn't, corresponds to a better leukemia-free and overall survival. And again, this is looking at monocytes, not the leukemic cells. We also noticed that there's a redistribution of cytotoxic memory and effector T cells, and it can be a surrogate for relapse-free survival, predicting fairly early who is going to do well. Particularly, I'd like to point out is that the patients who had redistribution of T effector memory cells to T effector cells did much better than those who did not. If you look at all patients, this holds up fairly nicely. Where induction of this transition results in improved leukemia-free survival, as well as improved overall survival. And in patients over 60 in this trial, very significant since we had less positive results in the original phase 3, we saw that this effect was even more marked in terms of predicting which of the older patients would likely benefit from this therapy after really only one cycle of treatment. So we had a pre-IND meeting with the FDA to get some guidance for a single overall survival phase 3 study for Ceplene IL-2 with an eye toward an NDA submission. We proposed a global overall survival study with Ceplene IL-2 that we expect to initiate in 2017 with 2-year overall survival as a primary endpoint. Ceplene low-dose IL-2 would be compared to IL-2 alone in order to satisfy one of the FDA requirements. We will look at secondary endpoints of survival, including LFS, at both 1 and 2 years. There still is no other effective approved therapies for AML following clinical complete remission. We've already shown that there is clinical efficacy in pivotal LFS phase 3 trial, as well as in a phase 4 follow-on trial. There's new biomarker data published in Oncotarget, presented at the AACR meeting in 2016, and a new patent has also been filed in February of 2016 for the development of Ceplene and checkpoint inhibitor combinations in lymphoma and solid tumors. And with that, I would end my presentation. Thank you for listening. Please get us any questions that you may have.